xenografts. She then sent specimens from each of those, samples from each of those to us. At that time, we were at MD Anderson. And we sequenced them in various ways as shown here. The starting point for this collaboration was fairly straightforward: to discover how culturing of glioblastoma cells might alter their genomic landscape, and then forward how transplanting neurosphere cells into a mouse might further increase or change their genomic characteristics. In this landscape figure of the 13 patients together with the neurospheres and the 3 xenografts for each case, the most frequently altered genes in glioblastoma are shown. And from a higher level, you might appreciate that by and large, there aren't that many changes when you take a glioblastoma tumor and culture it as neurospheres, and when you take those neurosphere cells and create xenografts. So we concluded relatively straightforward that these models quite well reflect their original tumor from which they were derived. At the same time, we still kept thinking about that extrachromosomal DNA. And we found that we could mine the whole genome sequencing data that we had generated from these cases to in fact predict extrachromosomal DNA amplifications. This is a zoom in of one particular case. The tumor sample TS is on top, and as you can see, this is the MIC locus very specifically. And as you can see, the MIC locus is wild type in the tumor sample. The neurosphere model has acquired an amplification of the MIC locus. And as you can see, it's kind of an odd amplification. It's a dual, there's 2 segments that are amplified at approximately the same level. And then we could also identify sequence reads that would connect those 2 segments to one another. In fact, we could create a circular path. So this suggested to us that this is actually not a straightforward duplication, but in fact, it's an extrachromosomal DNA. As you can see, the xenograft then also carried the same extrachromosomal DNA element. And even though I said the neurosphere might have acquired the MIC amplification, the alternative hypothesis, of course, is that this amplification was already present in the tumor sample, but maybe at a more low level, and the neurosphere, the culturing process of the neurosphere might select for those cells that carry the MIC amplification. And we are not able to distinguish those 2 scenarios. After we predicted that this amplification might be extrachromosomal, we performed a FISH analysis, interface FISH of the tumor and the PDX, but metaphase FISH of the neurosphere cells. And as you can tell from the red dots, specifically for the neurosphere cells, these are extrachromosomal. You can see that they do not overlap with the blue signal of the linear chromosome, the depistem. We then took the same concept and tried to predict extrachromosomal DNA elements across our set of 65 samples, and found that there's many, not only MIC, shown again here on the left, on the top, but also of genes like NDN2, murine double MINU2, PDGFRA, EGFR in many cases. And every time we used FISH to validate that these whole genome sequencing-based predictions were in fact correct. We found 2 of our cases, 2 of our 13 cases, showed a very particular pattern. This again is the order is shown as the tumor sample on top, the neurosphere culture as the second row here, and the 3 xenografts as the 3rd, 4th, and 5th row. This is a zoom in of the MET locus, MET, a well-known oncogene on chromosome 7. As you can see, both these original tumor samples, 3035 and 3077, contain a focal MET amplification that captures quite well the full coding sequence of the MET gene. You can also maybe appreciate that those are 2 different amplifications. We didn't have any contamination between the sequences, as the breakpoints are slightly different between 3035 and 3077. Now, and I'm not sure if you can see my mouse, but as you can see, this is the neurosphere, the 2nd row. And as you can see, there's no longer a detectable MET amplification in that neurosphere. But then when we analyzed the sequencing data of the xenografts, we found the exact same amplifications again. And we know it's the exact same amplifications as the breakpoint precisely aligned. So we concluded that there's a MET amplification in the tumor sample. that when we cultured those cells, for whatever reason, that MET amplification is outcompeted, cells containing the MET amplification are outcompeted by cells that do not carry the MET amplification. Maybe because the ligand of the cell surface tyrosine receptor kinase, MET, is no longer present in the neurosphere culture medium, which is the ligand is HDF. And maybe because when we transplanted these cells into mice, maybe that ligand was again present, and maybe therefore there was again a clonal selection process of those cells carrying MET, and therefore we see an increase and the MET-containing cells become the dominant clone again. So there's clonal selection of first MET cells, then MET wild-type cells, and then when you put them back in the xenograft, MET cells again. Now, when we then looked at the sequencing, the exome sequencing data we had generated of these same specimens, we found we had expected to see point mutations that would coincide with that dominant clone pattern. The order of the samples here is actually reversed. So the tumor sample is at the bottom. The fourth row is actually the neurosphere, and the three top rows are the xenografts. We had expected to see point mutations present in the tumor sample, lacking in the neurosphere, but present again in the three xenografts. And we didn't see that. And when we made that observation, we also concluded that the only way that we can explain that is because the mutations, which are residing on linear chromosomes, inherit differently from the extrachromosomal DNA, which is on a circular element that is not being segregated evenly. So that fits with this model where chromosomal alterations are on linear chromosomes are duplicated and inherit evenly between the daughter cells, pulled apart by the spindle complex at the end of telophase. Whereas extrachromosomal DNAs inherit randomly on the basis of where they happen to reside in that dividing nucleus and being encapsulated just physically with some randomness to it. So that means that when a cell divides, and this is just an example scenario, you may have cells that contain all the duplicated ecDNAs and other cells that have none. You may have cells where the ecDNAs are evenly duplicated. That's also, of course, a scenario that could happen by random chance, and every possibility in between. And when this scenario is true and the extrachromosomal element provides cells with some kind of competitive advantage, you might see very rapid accumulation of extrachromosomal DNAs just in a matter of a few cell cycles, which could lead you to, in this example figure, to having one cell that has many ecDNA copies. And again, if that cell is selected for, then this is the cell that's going to be the dominant clone in your tumor. Now, we found further evidence for this model, and this is actually unpublished data supported by the Brain Tumor Charity. We found further evidence for that model, because of course all the evidence we've shown so far is indirect and to a degree speculation. By counting the number of signals in a specimen cell by cell, so it's a single cell fish-based count of the number of copies of a particular fish probe effectively across a specimen. So in this particular tumor sample, we counted EGFR signals for a number of cells, and we counted chromosome 7 centromere signals and plotted them in this manner. And so you can see the EGFR copy number wildly varies across cells, ranging from as high as 50 copies to relatively few copies. Whereas the chromosome 7 signal is relatively stable. We may have some cells that are being duplicated, so you see a little bit, so you see 6 chromosome 7 copies, because we know in this particular sample, chromosome 7 has 3 copies. So the chromosome 7 signal, as expected, is relatively stable, but the number of EGFR copies varies wildly. We repeated that experiment for a number of other tumor samples, 4 in total, and saw the same pattern with some noise maybe, and again, the duplicating of chromosome 7 creates additional changes, but generally the variability in EGFR signal, which we showed by whole genome sequencing to be extrachromosomal as well as by the fish, is much different from the chromosome 7 signal, which is linear. We repeated that for the neurospheres, for 2 neurosphere cultures, and again saw the same pattern. And in fact, we reanalyzed some publicly accessible imaging fish data and found that that extrachromosomal signals show a significantly higher variability than linear amplified genes. And some of this data is also described in a bioRxiv preprint single-cell analysis by Kevin Johnson, who's a postdoc in the lab. Now, how more fitting than on the day where CRISPR was awarded the Nobel Prize to also include some of these slides, some work on our lab on the basis of CRISPR in the talk today. With one minor change, in our CRISPR experiment, we used a dead Cas9, which then allows guide RNAs to bind to a particular locus and to tag a particular locus without actually cutting it. And we combined the dead Cas9 and the guide RNAs with a fluorescent signal so that when the guides attach to a particular locus of interest, we can visualize that locus using the fluorescent tags. This method was first used, I think, by Stanley Chi and his lab at Stanford, and maybe reported in 2013. We took the same concept. We didn't use the exact same technology, but we used the same concept to try and tag specific breakpoints, namely breakpoints that reside on ecDNA, although there's no reason it wouldn't work for linear DNA. We leveraged this approach to tag ecDNA specific breakpoints, which we first discovered from whole genome sequencing and then validated using Sanger sequencing. This approach was put together and explored and implemented by In-Hee Yi, who is a postdoc in the lab. So again, we first mapped 4 specific breakpoints. Of course, this is a somewhat laborious approach, so we limited our experiments to 4 particular breakpoints at this point, all found in the same neurosphere culture, so that we could work with the neurosphere cells. And the neurosphere culture's name is HF3016. So we first mapped 4 breakpoints using whole genome sequencing, showed that these breakpoints are unique to HF3016 cells in comparison to the PC3 prostate cancer control cell line, where we do not see these breakpoints, and then used Sanger sequencing to find map the breakpoint, because whole genome sequencing can't usually find map the exact sequence that is at that junction. And of course, for our approach here, that's critical. Let's see. One other thing I wanted to mention here, because we're working with very specific breakpoints, we are somewhat limited in the length of the guide, and we're also limited to using just a single guide for tagging each of the breakpoints. Where often in similar visualizing dead Cas9 based experiments, you have the luxury of using multiple guides and thereby creating a very specific signal. We don't have that luxury because we're relying on the precise breakpoint. Therefore, this approach is going to be a little bit more noisy than what we've seen in previous attempts and previous experiments. So to demonstrate that this concept could work, we also used FISH, and of course FISH is done on metaphase cells, which are at that point no longer alive. So this is a reason why we can't use FISH to discover ecDNA behavior in living cells. But FISH, of course, is still very useful to validate this. So we designed FISH probes, again, mapping specifically to the breakpoint junction. And as you can see here, the yellow signal, the yellow signal with the breakpoint, as well as a sequence very close to the breakpoint, but not the breakpoint itself. The yellow signal here shows that this works. And here again, shown for the 3016 cell. We used a second neurosphere, 3177, which is derived from the same tumor specimen, but the recurrent tumor, but therefore containing the same breakpoints, to show that we can map, create breakpoint FISH signals, whereas PC3 has only a very low background signal. We then implemented this and found that breakpoint FISH, and then used again breakpoint FISH to validate that the guide RNA tagged with fluorescent molecules can indeed tag that specific breakpoint, and that is shown here for all 4 of the breakpoints that we attempted to tag. So we can see a very close proximity of co-localizing CRISPR signal with the breakpoint FISH signal. validating that this method works. And then just to relate it back to the original FIS experiments that I showed just a few slides ago, breakpoint specific guide RNA captures that similar ecDNA copy number diversity across cells as we had seen in fish, with much greater variability for the 4 ecDNA breakpoints in comparison to chromosome 7 and MUC4, which was our second control in these experiments. And then here's the key experiment that we initially have now used this technology for, which is to show that when you track ecDNA signal over time, which we now have the ability to do because we have ecDNA break junctions tagged in live cells, to show that when you have a cell highlighted here that's undergoing mitosis, here is the cell pre-mitosis, now it's entering mitosis and starting to segregate. And here we have 2 daughter cells, and as you can see, at the end of that mitosis, and once the cell starts to enter interphase, the number of signals between the 2 cells is wildly different. The top cell here has much more signal than the bottom cell. You might ask, why do we not see ecDNA signal here? Are we looking at actually de novo genesis of ecDNAs? We don't think so, because at this phase, the DNA is highly condensed, and we think that the guides are not able to access the DNA well enough to create fluorescent signal. So that's a potential limitation of the approach. Again, we're somewhat hampered by the use of just a single guide in our implantation. But there's no doubt that the 2 daughter cells here have a very different number of ecDNA molecules. Just counting by eye, you can easily appreciate that. So we then counted the number of ecDNA copies between the 2 offspring cells over a series of cells. And as you can see here, typically or often this is a random association. So the number of cells in daughter 1 is not in any way predictive of the number of the number of ecDNA signals in daughter cell 1 is not predictive of the number of ecDNA signals in daughter 2. Because there's random segregation. Whereas for our 2 controls, chromosome 7 and MUC4, we see a very strong linear correlation as expected, because these 2 controls are segregating evenly, so the number of signals between the 2 daughters should always be the same. So to our knowledge, and this is always a dangerous thing to say, but this is maybe the first direct evidence that uneven segregation of ecDNA is actually really how ecDNA is being inherited. So we submitted a manuscript describing this work just by coincidence earlier this week, and I don't want to claim that contributed to the awarding of the CRISPR today, but there's an interesting correlation there, and that of course is a joke. The drawback of presenting virtually is that you don't get an audience response, so I want to be explicit that that's a joke. Another observation we made, and we've made others that are more described in detail in the manuscript that I didn't include here today because of time issues, is that when you track ecDNA in live cells, you can also see that over time, the number of ecDNA signals can actually change rather quickly, and that may be expected just because of the competitive differences, but also the way they inherit, excuse me. So for this cell, we have a doubling time of just a little bit over 3 days, and we counted ecDNA signals over up to 7 days. And as you can see, there's somewhat of a decrease in the number of ecDNA signals of these 2 particular breakpoints, whereas these, there may be more of an increase. The key point being that it varies rather quickly between just a few cell cycles, whereas the signals for the linear controls are very stable. So we're really excited about this technology, and are going to use this now that we've extensively validated this. We're definitely going to use this a lot more for additional experiments. Now, up until now, cytogenetic studies and also databases like the Middleman database of chromosomal variants have implied that extrachromosomal DNA and double minus is a rare event in cancer. This 2011 paper, which scored cytogenetics, scored karyotypes of a large number of Samples reported that the overall frequencies of double minutes is 1.4%, which, if that were true, then ecDNA is actually a fairly rare occurrence. Now, as I described before, we were able to use computational analysis of whole genome sequencing data to detect and predict essentially circular DNA elements. Vineet Bhavna, who is a collaborator and a professor at UC San Diego, used that concept and developed the Amplicon Architect tool. In this recent paper, and I think they've now renamed the tool Amplicon Reconstructor, they show how you can use a seed region to determine a graph that can then be related to being circular. If you follow the steps of the figure, you can sort of understand the sequence of events that the tool, the sequence of steps that the tool takes to arrive at a conclusion of a amplification being linear or an amplification being circular. So in working with Vineet and also with Paul Michel, a Ludwig Cancer Center professor also at UCSD, we then used the Amplicon Architect tool to perform a pan-cancer analysis of circular DNAs. So we took all the available whole genome sequences from the TCGA and all the available whole genome sequences in the PCOG set, which includes most of TCGA, and computed in the cloud we first detected amplifications across that entire set of 3,200 tumor samples, and then computed in the cloud via the ISB cloud for TCGA samples and one of the Amazon clouds for the remaining PCOG samples, whether that amplification was either circular, whether it was derived through a breakage fusion bridge cycle, and it so happened that Vineet had earlier on developed a method to detect breakage fusion bridges, whether a amplification was heavily rearranged but linear, or relatively simple and linear. So we distinguished 4 classes of amplification. And then we classified the set of 3,200 tumors according to those 4 classes of amplification. And if a tumor included both a circular and a linear amplification, we would classify it as being circular, and so forth. So following that approach, we detected at least 1 extrachromosomal DNA in 14% of the entire cohort of 3,200 tumors. So about 10-fold higher than what was found by the karyotype analysis. Glioblastoma was the tumor type with the highest frequency of extrachromosomal DNAs, followed by sarcoma, esophageal cancer, ovarian cancer, but also major tumor types like breast and lung contain double-digit numbers of extrachromosomal DNA. We also found that in cases with relatively simple karyotypes like leukemia, AML, and thyroid cancer, we found 0 extrachromosomal DNAs. This also suggests that the approach we've taken here is likely very conservative, because there's plentiful literature reports in AML with extrachromosomal or double minutes of MYC. MYC is amplified with some frequency in this tumor type. Even though we analyzed more than 100 tumors, we did not detect any extrachromosomal DNAs in leukemia. Again, suggesting that that we took a conservative approach, and that the percentage of 14% is really lowballing the actual number. In previous analysis, we also found that in GBM, the frequency is actually more close to 80% or higher rather than 56%. Nonetheless, 14% of all cancers, of course, the data set we analyzed not being a total random representation of cancer in the population, but still, we think it's a very significant number, and certainly not as high as being reported before. This work was published just 2 months ago with Hoon Kim in my lab as first author, together with Nam Yen, who was a postdoc with Vineet and now a scientist at the Boundless Bio company, as a co-first author. So We not only predicted the frequency of extrachromosomal DNA across cancers, but also looked at the locus of where extrachromosomal DNAs were residing, finding that genes such as MDM2, again, CDK4 were frequently extrachromosomal. A bit less frequent, they were either BFB or heavily rearranged. EGFR, this is the MYC locus. MYC is frequently extrachromosomal, but also frequently heavily rearranged or linear. Same for EGFR. CDK4 and MDM2 are probably the most definitive outliers in terms of being having been preferentially extrachromosomally amplified. This is the same data, but now represented gene by gene, and you can see my point that I just made with MDM2 and CDK4 being highly frequently extrachromosomal, followed by important oncogenes such as EGFR and MYC. But as you can see, most oncogenes that are being activated through amplification are extrachromosomal in some cancers. Importantly, we also found that genes on extrachromosomal DNAs are expressed more highly than you could predict from their copy number level alone. So this analysis is showing copy number level compared to gene expression. Copy number is very well known to more or less linearly drive gene expression. In other words, more copies means two times as many gene copies means 2 times as much expression. But for circular DNAs, this linear correlation exceeded the expectation, and this may have something to do with the open chromatin structure and other properties of being a circular element that makes it more easily accessible for a transcriptional machinery. It also re-emphasizes the functionality of extrachromosomal DNA. And then finally, and importantly, outcome of patients with an whose tumors carry 1 or more extrachromosomal DNAs is worse compared to patients whose tumors we did not detect extrachromosomal DNA. And this is critical. This is independent from tumor type because, of course, there's an overrepresentation of sarcomas and glioblastomas in the ecDNA category. But when we consider tumor type as a variable, we still find a very significant worse outcome for extrachromosomal DNA. I'll speculate that the uneven segregation of extrachromosomal DNA has something to do with this, because it would enable cells to circumvent any bottleneck effect, for example, from therapy or from hypoxia that exists in glioblastomas. It gives cells a higher level of plasticity and flexibility to deal with these pressures that occur in the microenvironment, thus driving the worst outcomes that were shown here. So I'm ready to conclude and to summarize, first of all, that ecDNA contributes to intratumoral heterogeneity as reflected by the FISH data I showed you, but also the DETCAS9 tagging data. There's just a higher level of intratumoral heterogeneity because there's a variation in the number of copies from cell to cell of these extrachromosomal DNA elements. We now also show very directly that intratumoral ecDNA heterogeneity is driven by an uneven segregation during mitosis, long speculated and now definitively shown. ecDNA elements as a result, may rapidly accumulate, which enables a competitive advantage for cells loaded with oncogenic signals. And then finally, these are not rare events. These are frequent, they're commonly found in cancer, and they drive poor outcomes. And glioblastoma, as a tumor type, we found has the most high frequency of extrachromosomal DNA of all cancers. Finally, I want to acknowledge, of course, the lab that, in particular in Yi, who has developed the DETCAS9 tagging technology that I showed you. Hoon Kim, who did much, if not all, of the computational analysis of the 2018 and 2020 paper. Other lab members for their contributions, Paul and Vineet at UCSD for their collaborations, particularly on the pan-cancer study, and Albert, who's a faculty, an assistant professor, faculty member at my institution here, who was our collaborator for developing the DETCAS9 tagging mechanism. And then finally, and of course not least, the funding organizations, including the Brain Tumor Charity. So I'll stop there and try to address any questions that might exist. Thank you.
Thank you, Roel. Really interesting piece of work, which has so far stimulated a number of questions, which as I was rolling in, I was thinking, great, we've got a number of questions, and then you answered half of them. So I guess that shows a good presentation when the questions come in. It's a next slide answer. Maybe we can start with a technical question about the location of the ecDNA and why are the spots located peripherally in your fish?
I don't know that they are always peripherally in the fish, and it's not an analysis we've done specifically to show that they're inside the nucleus, that they're peripheral. It might be, if that were the case, there might be some functionality to it. We haven't really considered this yet. We have found, and this is not something I showed today, but in other experience, we have found that ecDNAs tend to cluster, and maybe the physical properties of clustered DNAs could potentially lead them to be more likely to be peripheral. But again, it's not a formal analysis that we've done, so I don't want to say that the ecDNAs are typically peripheral in a cell nucleus.
Okay. Something that's come up a couple of times in the questions, at least, is with regards to the downregulation that you can see of ecDNA, and particularly in vitro. Can you maybe comment on that, and can you comment on any implications that might have for the way that we do drug discovery, and any suggestions you have on ways we may be able to do that better?
Yeah, let me flip back my slides because I think it will help to address that question. So this is the slide that I want to go to is this one. So, or maybe even let me use this one. I think it's as clear. So we detected ecDNAs. So remember, we started with a set of 13 patient samples and then derived neurospheres and then derived xenograms. So 13. Across the 13, we found at least one extrachromosomal DNA element in 10. 10 of the 13 carried one or more extrachromosomal DNAs. All in all, we saw that the extrachromosomal element was lost in 2 cases, which was the 2 MET examples. So when we took the tumor sample and cultured the cells, 2 amplifications were lost. But they were actually still present, just at low frequency, because when we put them in a mouse, they reappeared. So they weren't truly lost. They were still a minor subclone among the neurosphere cells that contained the MET amplification. So the point being, I'm not convinced that extrachromosomal DNA is truly, that there's a dominant pattern of extrachromosomal DNA being lost in tumor cells, and when you culture cells. I certainly think it can happen. And we've taken the same cells that, some of the same cell cultures that are shown here, and passaged them up to 30 times. And certainly sometimes you see that the extrachromosomal DNA is lost. But just as often, we see that extrachromosomal DNA is maintained. I don't believe that there's a general pattern of of cells in vitro shedding these elements. Another way to address that is in this figure. So on the left, and can you see my mouse or not?
No.
Okay. So in this figure at the bottom, you see a blue dot chart that says ecDNA genes, and a blue dot chart that says linearly amplified genes, and ecDNA genes as a red dot chart. So these are ecDNAs detected in long-term cell lines. Cell lines, like some of the names are shown here, like CA718 or MB411FH, PC3. Now, PC3 was derived in the early 80s. It's a cell line that's 4 decades old. We can clearly detect MYC amplification and extrachromosomal MYC amplification in PC3. It's just one example of a long-term culture that has maintained its extrachromosomal DNA element. I think extrachromosomal DNA can be quickly acquired. It can be quickly lost, and that's especially apparent in vitro. So the way to leverage this in drug discovery experiments, and certainly we are pursuing those as well, is that you have to frequently check whether any observations hold true in terms of the ecDNA status. You just need to keep going back and do that breakpoint PCR or some other, DEPI stain or a FISH test to convince yourself that whatever result you're finding is truly derived because there's an extrachromosomal DNA element.
And I guess the second part of the question was around the drug discovery part. Are there any implications on that for the way that we currently do drug discovery, and any ways that you might think could be more appropriate?
Yeah, I think the implications are huge, actually. Work by Paul Michel in 2014 in a science paper showed that when you target GBM cells with that carry amplified EGFR with an EGFR inhibitor, those cells will initially respond. And if you look at the nitty-gritty, you find that these cells contained extrachromosomal EGFR, and on treatment with an EGFR inhibitor, that extrachromosomal DNA element largely disappears. But taken off drug, just as quickly the EGFR amplification came back. So the inhibition only worked for as long as that tumor cell was on the drug, but taken off, it quickly came back. And so you have a mechanism for very rapid resistance to the efficacy of the treatment. So targeted inhibitors are potentially not very effective when the oncogene is extrachromosomal. That's one point. Second point, because these elements are circular, because they inherit unevenly, because there's a wide range of biology of extrachromosomal DNA that we totally do not understand yet. I'm personally pretty convinced that there must be mechanisms that are ecDNA specific. For example, again, for replication of ecDNAs, to me, it seems intuitively unlikely that it uses the same mechanisms as for linear DNA with polymerase and all that. There must be, I think there's likely a different way that ecDNA replicates. And if we can understand that replication better, we may be able to design drugs that prevent that replication and thus have an ecDNA specific drug. It's another way, I think, to get at these tumor cells without hurting the normal cells.
That's the question about role for targeting ecDNA, so thank you for that one. There are reports of ecDNAs being expelled from cells. Is that happening in GBM? If so, what sort of vesicle are they in, and do they go on to be taken up by other cells? If so, is that just tumor cells, or is it some of the microenvironment cells as well? Is there any evidence around that?
Yeah, it's a really great question. So what that refers to is a concept otherwise known as lateral gene transfer. And you could conceive that that these elements leave the nucleus, enter the cytoplasm, and for example, a micronucleus, micronuclei exist in cytoplasm. That's well known, and they typically contain damaged DNA. And ecDNA may be a type of damaged DNA that gets enclosed in these micronuclei. And maybe there are ways for micronuclei to leave the cell in some sort of vesicle and maybe re-enter a different type of cell. One thing we think we can use the fluorescence tagging technology for is to study that exact question, to track, if you co-culture cells, to track if ecDNAs indeed leave the cell in some way or form and enter new cells. We haven't finished those experiments yet. I think there's some promise, but it's too early to really speculate too wildly on that. I think there's hints in literature. There are papers in literature. I remember one paper from MGH. I think it's a Nature Cell Biology paper that shows that MYC can leave and re-enter cells in certain circumstances and get sort of absorbed by the new cells. So this is a paper that's a little bit older and doesn't specifically mention ecDNA, but certainly MYC is frequently found in ecDNA, so it's conceivable that that's actually an ecDNA mechanism. Anyway, that's all speculation and hypothesizing to some degree, but I think there's some likelihood that there's lateral gene transfer. How and why, and if that enters non-tumor cells, no idea.
You mentioned that that the chromatin is open. on ecDNA and more accessible because of that. The DNA is more accessible. Do you have a hypothesis as to why that might be?
Well, ecDNA, of course, is a mechanism for activating oncogenes. So that by itself might explain or provide some explanation of the open chromatin status. The mechanisms of how that chromatin gets shut or opened and why that's different, potentially different for ecDNA, I don't really know. So I can't really say too much about that. The two most relevant papers, I think, that have some insights there are Paul Michel's Nature paper from almost a year ago now, where he and his lab show that ecDNA is circular and that it's all open chromatin. So it's a November 2019 Nature paper. And then there's a cell paper from Pete Scott-Sherry's lab around the same time that shows how enhancers are co-amplified and that they're part of the ecDNA structure. And because they're part of the ecDNA structure, the enhancer can mitigate expression of the cargo oncogene. So it's another, I think, piece of evidence or part of the mechanism of the very high expression of ecDNA.
I've got a couple of questions about the replication of ecDNA. Maybe you can comment upon the timing of that, whether that happens in time with the cell cycle or outside of that, and how they're maintained through that mechanism.
Yeah, it's a very good question as well. I don't know that we have the answer to that. Again, it's something where we think the tagging methods could be very useful to help address that question. It might help us to show replication. Of course, I also showed that during mitosis, the DNA appears to be so condensed that the guides aren't able to access, so we're not able to show the signal of a replicating ecDNA. That's maybe a technical hurdle that I hope we are able to address in the future. So we don't really have any specific knowledge or evidence on how ecDNA replicates at this point. And that's certainly one of the first questions that we're hoping to address, because I think that's going to be potentially very useful knowledge in developing ecDNA specific therapy.
And again, a group of questions which I'm going to interpret as whether or not these could be used for circulating biomarkers. So have they ever been found in things like exosomes, circulating tumor cells, or indeed just as circulating tumor, a form of circulating tumor DNA?
Yeah, I know my answer may appear a bit, my answers may appear a bit evasive, but it's really reflecting a lack of knowledge that we have around extrachromosomal DNA. So let me just again hypothesize to say that, you know, within a cell you can see how many ecDNA copies there are. Just look at the fish images on this particular screen. You can see that most cells contain, you know, dozens of ecDNAs. We know they can leave the cellular micronuclei. So it's conceivable that, and we know that circulating DNA is basically DNA released from dying tumor cells. Now, if there's a ratio of 50 to 2 of ecDNA, number of ecDNAs versus number of linear DNAs, it seems conceivable that ecDNAs are somewhat preferentially found among circulating DNA. But it's not an analysis we've done at this point. And it, yeah, certainly could be very useful.
Again, one for you to speculate upon, or maybe you have the actual mechanism for this. A number of the ecDNAs in your analysis contain sequences from multiple chromosomes. Do you have any speculation on how that's happened mechanistically?
That's a great question. So in our pan-cancer study, we found a wide diversity of genomic properties of ecDNA elements. So in our pan-cancer study, let me see if I can just quickly flip back to that slide. So we found in total close to 600 different ecDNAs in our set of tumors. So among those 600 ecDNA structures, we certainly also found different genomic properties, like microhomology varied between sarcomas and GBMs, and the level of microhomology at the breakpoint ends, at the breakpoint junction. What we also noticed is that chromothripsis as a pattern was more present in some tumor types than others, and it's associated with some extrachromosomal DNAs, but not others. So there's very simple ecDNAs consisting of a single segment that got circularized by ligating the tumor, the segment ends. And there were ecDNAs containing dozens of segments, including from different chromosomes. So I would say for that second, it's likely that there's an ongoing evolution. Maybe there's multiple chromothriptic events of shattered chromosomes that are being re-pieced together. And then via selection, the number of copies gets increased, because chromothripsis in itself is not a mechanism for amplification, it's just a shattering. So chromothripsis can create the original structure, and then again, via selection, you might get that structure as an amplification. But it's not the only mechanism that can create ecDNAs, because we definitely also see very simple single segment molecules.
One final question, and we've now obviously hit the witching hour of 4 p.m. British summertime, so we'll just ask one more before we close down the webinar, and that is there a connection between line 1 activity and the ecDNA production?
That's an interesting question. We haven't looked at that specifically. And maybe as a closing argument, again, it may seem like I'm evasive. I'm not trying to be evasive, but there's just not a lot known about ecDNAs. I might encourage all those that have dialed in today to think about these types of analysis, because we're not going to do all of them for sure. And line 1 is an interesting retrotransposon, not one that we are currently studying. So would certainly be very interesting if that type of activity matches the presence or absence of ecDNA.
Thank you. And I think, I'd echo that. I think that this seems to be an area which will only get more important to particularly GBMs and brain tumors, but no doubt other tumors as well. So yeah, certainly the more work in this, the number of those questions that Roel's had to speculate upon, we can maybe get some real answers to. So I wanted to take this opportunity to thank Roel again for his time and his efforts in this space, and thank you for the work you're doing as part of the charity and in the field in general. Thank you to everyone who has been here today listening to this. I'm glad that these webinars continue to attract a good audience and continue to attract the interactions that we've seen today. So thank you all for that. The next webinar that we'll be hosting is in a couple of weeks' time, and that is from Ruman Rahman and Stuart Smith, both of whom are from Nottingham, working on the hidden gene signatures in infiltrative GBM cells. The sign up for that will start soon, but please, if you have any comments, any feedback that you want to give to us about the webinars or any ideas for new topics, then please do get in touch with the team on the email address that you see at the bottom there. But until next time, wish you all safety in these challenging times, which seem to be getting worse at the moment, particularly here in the UK. But yeah, we look forward to seeing you all again in a couple of weeks. So thank you very much.
Thank you.